All right, I think we should start. So, good morning, or afternoon, or evening, depending on where you are. Um, so, this is the first of these sessions. I plan to give about 7 of these, going over various steps that need to be taken for curation overall. So, it's not. I'll focus on SNP array data, but this can actually be applied to a lot of genotypic data. If you have any questions, please feel free to ask them during the sessions. I'll see if I can find a way to keep an eye on the chat too, if somebody asks a question over there. Other than that, one more practical thing for the next sessions is that this weekend in the US we'll switch to daylight saving times. That won't happen in Europe, so at that point the time difference between Europe and the US will change, and that will mean that for the next 2 sessions at least, in Europe they'll be 1 hour earlier than today. So, but I'll also send an email to remind everybody about this. So I guess that probably works better for the people in Europe. And then otherwise, I have some slides prepared, and for today it'll be a lot of introduction and why do we need SNP curation, and I'll switch to the full presentation soon. And otherwise, I'll be showing things in the software itself and in the files. Those steps will also be in the presentation, but I might not necessarily show all the slides as I'm doing that, but the slides then serve as kind of a backup for you to go over them later. All right, so let's start. Or is it here? Oh, come on. All right, so first things first, and maybe I shouldn't have gone to the full slides yet. For software, I hope everybody was able to, and I'm going to switch back so that I can show you what I mean. Sorry about that. I hope everybody was able to get the software on their computers. If not, please let me know. So the software was divided in a group of software that needs to be installed, but can just be straight copied to your computer, which was in the copy to PC file. And so that's 3 files, or 3 pieces of software. One is Assist, the other is the FlexQTL data prepper, and the last one is Blink. So if you just copy those to your computer to a location that you know, then you should be good to go with this software. Then as for the other software that needed to be installing, I hope everybody was able to install GenomeStudio, Notepad++. The reason I recommend that software is it can handle large data sets, large amounts of data, and unlike WordPad or Notepad that normally come pre-installed on your computer, they tend to crash or really don't take a large amount of data very well. And then there was R and RStudio. So most of the time when I work with R, I work through RStudio, but when you install RStudio, it doesn't install R for you. So I hope that worked well for everybody. If not, again, please let me know, and we can see what we can do. And then the last part was Visual FlexQTL and the redistributable. That one, number 6, is only needed to run Visual FlexQTL on later versions of Windows. And that is somehow something went missing in the later versions of Windows, and if you don't install that, then you'll just get a blank notification box while running FlexQTL. I will come back to that once we actually will be using Visual FlexQTL, which is only in a few sessions from now. But I wanted to make sure everybody has all the software ready when we start with this. Now, don't know if there were questions about this, but I can quickly show you. So if you have a Visual FlexQTL installed, and I'll open Visual FlexQTL, then Visual FlexQTL uses R to generate some figures, and it uses the FlexQTL to run a bunch of analysis. And so we need to guide it to where that software is. And so that's done by, I hope everybody can see the yellow circle around my mouse so that you have a better idea of where I'm clicking and where I'm going. So under the Tools tab of Visual FlexQTL, here in the top left of my screen, you choose Settings under Tools. And then here, as for me, it's already all filled in. But what you need to do is find where you installed R and find the R script software, or the R script executable. So you do that by pressing these 3 little dots, and then a window should open and you can start looking for it. Normally it's under Program Files, R, then whichever R version you're using, and I might have to update this, and then bin, and then you can find the R script, and then you can double click on it, or click on it and then click on OK. And the same goes for the FlexQTL and PostQTL, that you need to look for the FlexQTL console.exe and the postqtl_console.exe, which is, I'll quickly say it. In my case, it's under in the Windows or the C drive, then under Program Files, but the x86 one folder, then Visual FlexQTL, and then I chose the x64, because I think there should also be a Windows 32 which also houses it, but I prefer this one because most computers right now will use this one. And then as for the license was given on Dropbox. It would be in the software folder. So if you in the software folder, there would be a FlexQTL.lic or LIC, and that's the one you need to show FlexQTL where it is. This is a license file. This is the one you got is only a temporary one that will expire the 1st of July, I believe. Those who are working within RosBREED normally should have their own. And if you want to use FlexQTL more, I suggest you contact Eric van de Weg at Wageningen University, and he can help you get a license. They're free, but they want to keep track of who's using their software. And then as for figures, FlexQTL generates a bunch of figures. So if you're back in FlexQTL, still in settings, so we were in the general tab. If we go to the output tab, I think standard, this is 800. I changed it to 1200. This makes the figures a bit bigger that are generated by Visual FlexQTL. So that helps if you want to then later use those figures in a publication or in the presentation. So that should be most of the software setup. I guess the other one is R and installing its packages. For those who are not familiar with R, this is a bit of R script. In some cases, there's functions in R that are not standard in R, and one needs to have those installed in what's called a package. FlexQTL uses some of these packages, so if you need to install these, you go in RStudio. So if you open RStudio, you might not have an R script open here, but otherwise it should be look very similar. You don't need to have an R script open to do this. You go to Tools, Install Packages, and then this window should pop up. I think for most of you it will be set like this, where it says Install from the repository, and then you can search for all those packages in the list. I hope that worked all well for everybody. So you can do all of them at the same time. So I think by providing either a space or a comma. So if I put a space here and then I type _, and so then I can go add all these packages in this list. I'm not going to click on install, but if you click on install, it will install all these packages. And then there's one more package that needs to be installed, which is the PEDI haplotyper, which does not appear in this list if you look for it, but it is given with the software. But for that, you need to choose package archive file, because then you will look on your own computer for one. And so mine are in this folder, I believe under software, our packages. This notepad file gives another time the list of all the packages to install, but the PETI haplotyper package is in the PETI haplotyper folder, and then it's this file, the PETI haplotyper _1.0.tar.gz, and then you've selected that one and you click open, and then you can also click on install, and that will install that package for you. And so if you've done all that and that works for you, then you should be able to use all the software that we will be using throughout the sessions. And then the other thing I asked you to look at is Excel shortcuts, and I think I'm going to quickly go over them just in case, just to show you. So with the control, I hope you can see which cell is selected in Excel. If I press control, and push any error, my selection just jumps to the last filled cell. If I started on a filled cell, if I start on an empty cell and I click control and in this case up, it will jump to the first filled cell. That's just an easy way to move around your data set if you need to fast. Especially in this small example, it might not be super useful, but if you have 2,000 or 3,000 columns, then it's really easy to just quickly go somewhere or find some missing data. Now if you hold shift and you press any error, then you start selecting cells. So you start with the cell you first selected, and then whichever direction you go, you just add one row or one column as you go. And if you combine that with control, so now I'm holding control and shift, you are not only jumping to the last cell, but you're also selecting everything in between. So that's also very useful to know if you didn't already. What else? Oh yeah, so holding control, and that's not only in Excel, that will also be in some other software. If I hold control and I start clicking on cells, I'll just keep adding those to my selection, and so you can choose whichever custom selection you want. If you want to copy this, you need to have everything in the same column or in the same row, because otherwise you'll get an error message that you can't copy this. But you could color this, so you could do some other things with that. You could color these cells, for instance, if that's what you want to do. Then I guess autofill. Most of you, I think, know the autofill as this, where you just say, let's use a formula. This is equal to that, and then by clicking this little square here in the lower right corner, it just drags it all down. That works really well. But if you want to do this to the right, let's just equals this. If you want to fill in to the right, well, I think most of the time I used to drag, but instead of clicking on this little square and dragging it, I could also select all the cells, and if I click on Ctrl R, then it will fill everything for me that way. Again, for a small amount of cells, that is maybe not necessary, but if you have a lot of columns and you want to do that, especially with the large data that we work with, this can be really useful. And then I've been dragging and showing you the formulas, and for those, and in case you don't know, so if I say this formula just says this needs to be equal to cell A1, and I say drag this down, this will become cell B1, or A2, sorry. So it will keep, as I'm dragging it down, it will just follow this list here. And if I drag it to the other side, it will follow this. And with F4, by pressing F4 on a value, oh, why is that? F4, then you get these dollar signs, and dollar signs means that you lock either whatever the dollar sign is in front of in. So if I lock everything in and I drag this now, then I will always get the same cell because it will keep referring to that. And that can be useful, again, if you work with large data sets and you want to lock in some things. And then the final thing that I will use, and that maybe is known a little bit less, is the index match function. I'm just gonna 15, 3, 4. So in that case, if I would like to know which values of 1 in this column are linked to a 1 in this column for column A. So I want to get, here I would want to get A, here I would like to get D, for 6 I would like to get F, and so forth. Then there's a VLOOKUP function, but which maybe more of you are familiar with, but that requires a certain format of your data. For instance, the values you're looking at need to be in the first column. With index match, you don't need to do that, and the way the function works is you use the function index, and at that point you choose which column you want the data to be returned from. So in this case, I would like to know the letters, so I highlight column A, and after that I click on match. I use the function match, and then it asks the lookup value, which is which value am I looking for, or which value do you want the result to be returned for. And in this case, this would be the cell which has 1 in it, which is now hidden by the formula. And Excel needs to go look for a 1 or a 4 or a 6 in column B, so that's the next column. That's the next thing I tell in the function. And then I use 0 as an exact match, because if you don't, then there's a chance that a 4 and a 44 or a 3 or something like that, things that look very similar, are treated the same. And most of our time when we work, for instance, with seedling numbers, we want the exact seedling number and not something that looks like it. And so it's telling me I need to add another bracket, and at that point it will return me the values. And you see that 15 gives an NA because there's no 15 in column B, so it can't return a value for that. So that might happen when you're using this formula and you're using individuals' names and you made a typo, or the names are written slightly different in the column that you want to import the data to compared to the table you want the data is from. So if you're not familiar with these Excel functions, check those out again, because I will be using many of those throughout these sessions. Alright, and then I can switch to the presentation. So I first wanted to make sure that everybody has a good understanding of how where the data comes from that we work with, how the SNP arrays work. So for those who don't know, a SNP stands for a single nucleotide polymorphism. If you look here in the figure, you have 3 DNA sequences, and they're all exactly the same except for one nucleotide. There's a difference, so that's a SNP. So what are some of the advantages of SNPs? They're really common. So millions have been found when resequencing apple data or cherry data, or 100,000 at least. There's a lot of them in the genome. And they allow for screening of thousands of these regions, SNPs, at the same time. So that makes it easy if you wanna do some genome-wide genotyping. SNPs can make it really easy. So one of the tools that were made to do that are SNP arrays. And so one of the advantages of SNP arrays is that they screen thousands of loci, and they're consistent. The information they give you is consistent between individuals. So if I run 20 individuals of an apple or a cherry individual cultivar, just 20 cherry individuals, then you will get the same. You will get information for the same loci back every time. Whereas with, for instance, genotyping by sequencing, you might end up with a lot of missing data, because depending on the individual that you run the GBS on, the genotyping by sequencing, you might get information back for different loci. And so either if you want to combine that, the data over many individuals, you either end up with a lot of missing data or a strongly reduced data set. And SNP arrays, there's multiple platforms. The two most common ones, or the ones that with the crops we work with, are the Illumina Infinium array, which the Apple 8K and the Apple 20K, the Cherry 6K, and the new array with the add-on, which we will be using data from for these sessions, and also the Peach and the Peach array. They're all on this platform. So the sessions will also most focus on data from this platform because that's the platform I have data for. But there's another common platform is the Affymetrix, and so the Apple 480K, as well as the Strawberry and Rose arrays are made on that platform. And so this image shows an Illumina Infinium array. So you can see it's not that big, and you see here all these squares. So there should be 24 in total, and so in each square is where one sample is tested for the amount of SNPs that are on the array. So it's really small, the area in which you screen for all these SNPs. So it's pretty cool technology. And so I have a quick video from Illumina to show how the array works, so that again you have an understanding of where our data is coming from. I'll show it now. Sorry. Alright, I'm gonna stop the video here. The rest talks about GenomeStudio, and we'll come back to that later today. So, just a quick summary of what was said in the video. So these arrays have a microbeads, and each bead has many probes that targets a single region in the genome, and these probes are complementary to the DNA right to the nucleotide before the nucleotide where the SNP is. And so during the reaction, our DNA will bind to it, and then in the next step, a single base will be, this probe will be extended with a single base, which is the SNP, or the nucleotide where the SNP is at. And so these nucleotides are labeled, and so by looking which label was incorporated, we can see which the genotype is. So, and one other important thing to mention, at least, is that, at least for the Illumina array, I'm not 100% sure about the Affymetrix one, is that the A and T nucleotides are labeled in exactly the same color, and the C and G nucleotides are also labeled in the exact same color, which means that we cannot distinguish an A from a T because it looks exactly the same, and we can also not distinguish a G from a C. Now, the arrays are designed in such a way that we don't target such SNPs. So the polymorphisms we'll see, or with the SNP array, are always going to be AG, AC, TG, or TC polymorphisms. And so on a bead, here only one probe is shown, but there's many probes. And so when a homozygous individual, or the DNA of a homozygous individual binds, you'll only see one color, whereas as a heterozygous, then both colors we expect to see. And so that leads in GenomeStudio, and I'll come back to this again, but for the next slides you need to have a little bit of an understanding the output looks like. So this is the output for a single SNP, and it shows the intensities that were recorded for this SNP over many individuals, and each dot in this plot is a single individual. So you can already see that the individuals group in 3 clusters, as is expected, because we're expecting 3 genotypes. Now, on the y-axis is, in this case, is the total intensity observed, and on the x-axis is the proportion of the B allele. And if you wonder what the B allele is, GenomeStudio calls everything as A's or B's, and A's, I believe, are the A and T nucleotides, and B are the C or G nucleotides. So rather than saying this is an AT SNP and then next is a GT SNP, it calls everything AB. And so this is given that this is the proportion on the x-axis of how much B allele was observed. Then if you're really at the beginning at almost 0, that means no B was observed, and so this means that all these individuals are AA individuals. And if you go to the other side of the graph, you see a proportion of 1, which means almost everything that was observed was the B color, and so these are BB individuals. Whereas somewhere in the middle, where the proportion of B allele is 50%, that means that the other part was A, the other 50%, and so all these individuals are heterozygous AB individuals. And then the last thing I want to point out is that you can see 2 shades of each color, and GenomeStudio will only call individuals AA if they're in this darker shaded red area, and will only call things AB if they're in the darker purple. Anything that falls in between will be considered a no-call, and GenomeStudio says it can't call it reliably, or cannot assign a genotype, that is, reliably. Don't know why it's okay. So we're going to talk a lot about the curation of SNP data, but why do we even care about all the SNP data, and why do we care that it needs to be curated? So I'm going to briefly show 2 examples of what we can do with the SNP data. I'll come back to that in a later session too. But in this case, what you can see here is based on SNP data, and it's the whole genome SNP data of an individual cosmic crisp. So that's an apple individual, and what you can see here are 2 rings. I believe the outer, so because it 2 rings which represent its genome. You have 1 ring represents the alleles that and the chromosomes it got from its mom, and the other ring represents the chromosomes that it got from its dad. And so you see these darker black lines regularly. Those are the chromosome delimiters. So each section within is a different chromosomes, and in apple we have 17 chromosomes. So what this is showing is where each chromosome segment is coming from throughout the ancestors of this individual. It's also showing these diamonds, which are outcomes of our DNA tests, which are saying how well, predicting how well or how this individual will perform for certain traits. But to do this, we need to be able to trace all these alleles through the pedigree and the ancestry. And if you have errors in your data, that's going to be much harder than if your data is all clean. I believe also in most of these cases, segments are only shown when the segment is large enough. So once you get errors in that, those segments might be broken up in little smaller pieces, and they might not even show anymore. Then the second application that we've been using this is once we have a SNP profile for an individual, we can start deducing. So this is already SNPs that are combined into haploblocks. So these are different alleles throughout the genome, in this case on chromosome 10. And at this spot, the PG one is a DNA test that we're using to predict performance of an individual. It's a DNA so it's an SSR DNA test, I believe, that we use. But we can deduce what alleles it has for this DNA test without actually running the DNA test, because we have the SNP profile. But again, at that point, we need to have accurate SNP data, because if there's errors in it, we might come to a different conclusion and give different advice to a breeder. So for that, we need to make sure our data is right. So Is there a lot of errors in our SNP data? Well, yes. So I showed you earlier a plot that is a best case scenario. In a lot of cases, our SNPs don't always look that nice. They may hold really valuable information, but the automatic calling of GenomeStudio doesn't work. So in the first case, you see here 2 SNPs. In the one on the left, all the intensities have been moved to the left. So if you can see here at the bottom is 1, so we would expect the BB cluster here, but it's not. It's almost in the middle of the plot. In this case, GenomeStudio still recognized the 3 clusters correctly. Unfortunately, on this side, it didn't. So we can I think we as humans can clearly see 3 clusters, but GenomeStudio doesn't recognize them. So if you would just take this output from this SNP as it is, all these AA individuals, or most of them at least, are purple, which means they would have been given the AB genotype rather than the AA genotype. Sometimes we get additional clusters, or in the NAPL, regularly. And sometimes GenomeStudio calls them correctly, but in many cases it might not. This might not be an AB cluster, for instance. And so either we have to know that we can't trust this SNP and throw it out of our data, or we need to make our adjustments to make sure that whatever we get for genotypes are true. And then the other point I wanted to point out is that in the first image, you see one germplasm collection of individuals that was genotyped, and we can see 2 clusters, an AB and a BB cluster. Now, in the second set of individuals that was genotyped and that was run separately in GenomeStudio, there was only 1 cluster, and it was called the BB cluster. GenomeStudio, my experience, tends not to call, if there's only 1 cluster, the cluster heterozygous. So it will always try to make, if there's only 1 cluster, the cluster a homozygous cluster. Now, if we combine all that data for the 2 collections into one run in GenomeStudio, then we see that all these individuals are actually grouped together in the AB cluster. So they were not BB, they were AB, and so there is several issues with genotyping in GenomeStudio. And it's not just SNPs that have this issue. For instance, with our SSR data, the MA indel test, we have a 400 allele and a 402 allele, and so also in that case, we might call it incorrectly, and if we do that, that might have implications, because one allele is linked to low acidity and the other is to high acidity. So whatever we advise, we need to make sure our data is right, because if we make a mistake here, we might give the wrong advice. So it's not just SNP data that needs this. And not only for those those examples, there's other issues. For instance, with genetic maps, if you have errors, your genetic maps might not be right. And in that case, if you have 2 different sources of resistance, for instance, for a disease, and your maps are not correct, you might map those disease resistance loci at if they're actually the same, but you might map them separately, maybe 5-10 centimorgans from each other, and then you might think that you are able to combine both resistance alleles on a single chromosome through a combination, when it's actually you cannot. So you might start making breeding decisions or strategies based on false maps. So you want to get your data as clean as possible. Also, you might find that certain alleles or SNPs are associated with a certain trait levels, and again, that might not be true if your data is full of errors. And the last part about incorrect imputations, for instance, if you look at this little pedigree, we have here Gala, which is falsely scored as BB, and we notice that there must be an issue because one of the parents is AA and the other parent is BB, and the only way Gala can be BB if kids orange red would have a B allele, which is not the case. Now, if we look at the rest of the pedigree, we could deduce that Gala must be wrong, but not all our software knows that, and the software might actually think kids orange red is wrong, and give Kids Orange Red a B allele to make sure that Gaelic can be BB. But then at that point, there's still an error because Delicious and Cox are both AA, and how would Kids Orange then be AB? And so it might change either one of these to also have a B. And in pedigree, in our data, in RosBREED at least, that is strongly pedigree connected, you can see easily how one error can just start spreading through your data set. So you want to get rid of those errors as best as we can. And so identifying errors, well, we can start looking at every single SNP and see if everything worked well. But as you've seen, there's arrays with 480,000 SNPs. So do you really want to go and look at each individual SNP? And then also, we run these arrays on many individuals. So do you want to really go and check each individual? There's hundreds of thousands to millions and maybe even billions of genotype calls. So it becomes really hard to just go and check everything one by one. And so at that point, we need a workflow that can help us identify the issues and identify on which regions we need to focus. And so this is the workflow I'll be talking about starting today and over the next sessions. This is a figure from a paper we submitted. And so there's different steps that we'll be talking about. And so today I'm mostly going to talk just about GenomeStudio and getting our genotypic data. And then from the next session onward, we'll start talking about fixing issues with our data set. All the way up to the last session where I plan to talk a bit more about what we can do with the data. So as I said, that figure came from a paper that was submitted at the end of last year. I think about a week ago I got it back with some minor editorial revisions, so nothing big. The pipeline passed the review process, and the paper is already available on bioRxiv if you want to read it. I think I sent that link before, so I hope you had a look at this already. So then let's have a look at the data and how our data works, and I'm going to go back to the actual files now. So I don't need this anymore. So, nope, sorry, wrong folder. Here it is. So if you look in the SNP curation sessions, I asked you to download the 6+9 sweet cherry data. So that holds the data as we receive it from the iScan facility. So if I double click on that, we can see two folders. These are dates. So these are the dates I received the data. So you can see we got our data in on 2 different dates in 2018. And so each folder contains the data for multiple arrays that were run on during that date. So if I look at that, then we see that there's multiple folders within this. And so each folder are the results for a single SNP array. So normally that's 24 samples that are in each folder here. And then we get another Excel file, and I'll come back to this shortly. And this Excel file lists how the samples were distributed over the different arrays. So you see each array has a number, and if I open this folder, well, as you can see in this folder, there's not a lot of individuals because I need to make sure I can only share the data of specific individuals with you. But you can see here that in this case, there's a bunch of files that have R02CO2 in them, and I think that's on the next slide. So that means that this is the data for row 2, column 2 on the SNP array, and for that you need to think like this, or maybe the other way around. But there's 12 rows on a SNP array and 2 columns, and so these are like from here all the way to here are all the results for a single sample. You see red and green because those are the 2 colors that are used to distinguish the alleles. And then all this is the results for another individual. And so then if I look at the sample sheet file that comes with it, I believe I always get both the CSV and an XLX. If you don't, then if you only get a CSV, I'll come back to it, but make sure you save it as a full Excel file, because if you might have some troubles afterwards if you don't. So I opened one of these files, and this is what a file looks like. So it starts with a header, which gives some information on the blade runs. Often this is blank, but you can fill it in yourself if you then want, so that you can keep track of who run this data, which project was it under, what is the actual data. Then the second part is a note about what needs to be below, which data should actually be available in this folder or in this file, and which is optional. Then the manifest link says the name of the file, and that file is also in the data folder. It's this file, so these names should correspond to each other. And this file lists how the SNPs, I believe, how the probes for the SNPs are distributed on the array, so that the software knows which little intensity dot is what array, what probe, and so which locus you're testing for. So this differs between different array types. So the 8K array for Apple would have a different one, and the Cherry 6K array has a different one than this, but normally they should all be the same for every 6 plus 9K array that is run in Cherry. And then the last part is the data part, and this tells you, well, a sample ID, which gives you the name of your individuals. It tells you the Centrix barcode, which is, if I open this folder again, this is really just telling you which is the number of the array, which array was this sample run on, and then the position is telling you where on this array was this run. So this is these three: a sample ID, the array number, and the array position. Those ones are the ones that you absolutely need in this file. Any other column is optional. So for instance, we have a column here that's saying on our 96 well plate, where was the sample located at. It's also here chip 1 to 4, so it's giving these arrays a different name. You can see that there's a column for replicates and parents. Most of the times when I get it, this is blank and I have to fill this in myself, but so you can give in parent information in here, and if you have replicates, you can give in replicate information in this column. You need to make sure that if you do that, slightly bigger, that whatever you fill in here matches the way you write it in here. And I'll go back to the slides now for a second to show you some other columns. So I'll make this big so everybody can hopefully see it well. So these are some other examples of data sheets. Some is from when I was still working in Belgium. All my individuals had a code, so the sample ID, so everything was coded, but they also had their actual name. So I would put in both. In here, I was able to, and we'll talk about this, I think, in the next session, with the SNP array, figure out what the ploidy of my samples was. So I added that as an extra column. And then later, when I start working in RosBREED, I also compared both data set and marked if individuals were in RosBREED. Just in case something was wrong with some of the SNP data in RosBREED, I could go back in this data file to this data set and see if that hold any valuable information, which it actually did. Then in RosBREED for Apple, with the Apple data, I also added, for instance, CRS, which means crop reference set, which tells me that all these individuals can be made publicly available within RosBREED. There's also breeding pedigree sets, which are proprietary to the breeding program. So then these would have BPS instead of CRS. And here's a bunch of extra columns saying then to which breeding program it would belong if the data is not publicly available. So there's a lot of extra columns you can add to this to keep track of your data, and GenomeStudio will, so the software that does the genotyping calling, will I'll import this. It won't know what it means, but you will have it in GenomeStudio in case you want to do some filtering based on that. I think I forgot to mention one thing. Yes, so this array number sometimes appears as a scientific number, like it's shown here in the slides. The issue with that is if you save this as a CSV file rather than as a full Excel worksheet, that sometimes that it will remember this, but it won't remember anything that after what it displays. So it knows there's a bunch of zeros after this, but it doesn't know that there's actually a 1 here or a 2, and so it actually you lose the link to the actual array sometimes. So for that, I always save it as a XLS or as XLSX version of the file. I do all my edits in that kind of file. GenomeStudio does require a CSV file to load the data, to load this file. So after I do all the manipulations and editing of this file, I will save it as an XLS, but I also make a CSV copy. And every time I need to change something afterwards, I go back to the XLS version and not the CSV file. So yeah, and others examples, what you could do is you could add a source location if your samples come from many sources, or a tree number if you have many copies of a same genotype. You could indicate which one you took it from if you know. So you keep track of all that data in a single file. The other thing I would advise you to do is, if you look at the sample IDs, and I made sure in this file, but sometimes when I get this back, there's some typos in this, or there's people who couldn't read the handwriting. I mean, if they got some things that were handwritten, and so if you have an error here in how the individual is named, a typo or anything, and you start using that throughout all the rest of the workflow, that type will also spread. So make sure you check all this to make sure all you're in the to make sure this is all written correctly. And then secondly, some of the software we'll use doesn't like spaces or weird characters, and so I advise you to copy your sample ID, call it sample ID original or something else, so that you have how it was. But then make short names, or if names have spaces or weird symbols, provide another name for this individual so that you don't get issues later on running everything, because especially with spaces you might run into problems. And the other advice I have is when you get these data files, make a backup before you do anything. Save it somewhere else, so that if you do something wrong, you can always go back to the original files as you got them. Then one more note on the duplicates. I found out, so in this case I have Rainier, and we run 2 samples of Rainier. And here's just a bit of advice, is keep one just as Rainier. Don't call it replicate 1 and replicate 2, because that would mean that if you have Rainier as a parent, you would also always have to call the parent's Rainier replicate 1, and that might be hard to link it to existing phenotypic data or other things where the individual is just called Rainier. So keep one as Rainier, and then every other replicate add something like replicate or duplicate or something to it. Now this is a bad example because this has a space and it should not have a space. The second thing of advice is here, if you go into the replicate column, what I did first is saying, well, Rainier is a replicate of Rainier replicate 1, and Rainier replicate 1 is a replicate of Rainier, so I would give in both. What that does is in GenomeStudio at least, when it starts comparing replicates, it will give everything twice. It will say, well, I compared Rainier with Rainier replicate 1, and then the next thing what it will do is say, I compared Rainier replicate 1 with Rainier. Well, that's obviously going to give the same result. So there's no point in having this twice. So only the ones that you call replicate, I advise you just to refer to the, I guess, original or individual that isn't named replicate. And then for GenomeStudio to read all this data, the way it came here is that it came in 2 folders, and I have to, I will show you that shortly, I have to guide GenomeStudio to where all my data is saved. Now if I say this folder, then I will only get all the samples from this folder, and if I say this folder, I will only get all the samples from this folder. And if I say, well, it's all the data you can find within this bigger folder, 6 plus 9K sweet cherry data, at that point it will just look in this folder and it won't start looking in subfolders, is my experience. So if you want to load all your data at once, you're gonna have to well, get all the data in one folder. So for that, I would create a new folder. I'm gonna call it RosBREED, and you can follow along if you want, so that you can also load the data in GenomeStudio yourself. Might call this RosBREED all or something. And then I need to get all the data in this folder. So normally I advise copying it, and I think we can do it now because it's not a lot of data. But if you have a lot of data, let's see how quickly that goes. Okay, so that goes pretty quick. So then I advise you to copy everything so that you still have your original folders as well. And so if I want to select everything from the file, I hold Shift and I click the first and the last one, as we could do in Excel. And then I can copy everything into a single folder, and depending on your computer, that may take a little bit more time. So now we have all our data in a single folder. The second thing that we need to do is then get everything into a single sample sheet file, because that's what will be loading into GenomeStudio. So for that, again, I start with the XLS version, and if I open that, that's the first one, and I need to get the data of the second file, of second folder too. So I need to get this one, and if I open that, there's a lot more data in this one. So it's what you can do, or what you need to do is just get all of these columns pasted underneath all the data information, pasted under this data information, because the manifest will stay the same. So you just want to have one file that links all the data. So if anybody's wondering why it's colored, it was just for me to easily see which array the samples were on when I was selecting all these samples. So I select everything. So in this case I did it with Shift, but so I select all these columns. I have to make sure that the order of the columns is the same as I copy it, but it should be. And then I just copy everything in this one file. So this now houses all the data for the data that you have. So it holds all the references, or is telling for all the samples that we have where to find them. And now I'm going to save this as a new file because I want to keep the old file. I'm just going to call this all also. And importantly, I'm going to first save it as an XLSX file, or full Excel file. And then secondly, when I want to save this, I go back to File, I click on Save As again, and then as I go to Save As here, where it says Excel Workbook, I look for comma CSV or comma delimited, and I can just save it under the same name because it will recognize it's a different file type. So if I save it, and then depending on which version of Excel you have, it will ask you if you're okay with that, because the file will lose some of its features, like it will lose all the color coding once I close this and open this again. So if everything went well, you will have now one folder with all these SNP array folders, and one file or two files, one Excel file and one CSV file that refers to all the data. And so now we can go to GenomeStudio and start loading in our data. And to do so, well, we have to open GenomeStudio. And so, I normally don't get this, but okay. So, if you open GenomeStudio, probably for you, this will be empty. If this is the first time you use GenomeStudio. Otherwise, it will show you previous projects you worked on. And so to load in our data, there's two ways. So the first is if you go to the top here and you click on File, New Project, and then you choose Genotyping. And at that point, a pop-up window should appear. I'm going to cancel this because there's another way. If you can look here, more in the middle of the GenomeStudio, it also has this new project part of it, and it also says genotyping, so you can just click on this, and at that point the same pop-up window will appear. So there's two ways to get there. And so once we're in here, this will guide us through the process of loading our data. So the first thing it asks us is the project repository, which is asking us where do you want to save your project. Now this should probably be a location that you remember where you save your project. So in my case, I made a new folder called GenomeStudio, and since I already made a project before, it already holds a project folder, but I want my projects to be saved in this folder. Of course, it's up to you where you want to save this project and all the data for it. And then we need to create a name for our project. So, given that this is give it a meaningful name so that you remember what the data is. So in this case, I would say cherry 6 plus 9, 9 K array, and I could call it CR because it's the crop reference set, or it's data for the crop reference set that this holds. So once I've given it a name here and set where it should be saved, I can hit next. And then there's two options, and I always choose the first one, which is use the sample sheet to load sample intensities, which is the sample sheet is that Excel file that we just created with all our samples in it, and saying which part of the array holds which sample. So I can just go to next, and then it asks me, okay, so where is the sample sheet? And so I click on browse, and I should guide it through the sample sheet, and as you can see, it only shows up CSVs here, so I'm going to use this file that I just created, and I'm going to say this is the one by clicking on open. The next thing that I ask is where is the data repository. So this is not where is my data project going to be saved, but where is the actual SNP data. And as I said before, it asks, in my experience, it doesn't look in subfolders, and that's why we copied everything into one big folder. And so I'm going to tell it, nope, this is not the right one. So I made one in this folder, and I called it RosBREED all. And so this is the folder that contains all the folders with our SNP data. So I'm going to say this is the folder. Now the next one, it asks where is my manifest repository. So it's asking me where is the manifest, and the manifest was that weird named file that tells you where, or tells the software how the probes are distributed on the array and where to look for which probe and thus for which SNP. And that is this file. So that file in my case, and if you copy this folder as it is from Dropbox, it will also be for you in the 6+9K Sweet Cherry data folder. So it's not asking you to show the file, it's asking you to show where the file is stored. So in this case, we have to say which folder that is, and so I just have to say it's the 6+9K sweet cherry data folder. It doesn't show the manifest file here, but again, if I open the folder itself, we can see that yes, this folder, this file is actually here. So this is okay. So once you've defined all these, where all these three files are, then you can hit next, and then it gives us some more options about the clustering. It asks if we have a cluster, import cluster position. So that is if I've been working extensively with this array and I've already fixed a bunch of SNPs and I've set, so in the case where clusters are not in the position they are expected, I could manually change that in this software, and I can save that so the next time I run a project, it will know where to look for the cluster rather than starting from scratch. So I could check this if I have such a file. I don't have this for a 6+9K array, so I can't check it. I do want the clusters to be, the SNPs to be clustered. I wouldn't worry too much about this right now, because in one of our next analysis, or the first analysis we'll do with the data, we'll probably exclude some samples. We might do a few other things, and so then we need to recalculate this anyway. And then the GenCall threshold is set to 0.15, which is the standard. I haven't changed this. If you remember, let me see if I can quickly show this on this plot. This defines how wide or how big this dark shaded area is. If I make this, if I change this, this will change how far away from the main cluster it will still call things. And I believe the higher the value, the smaller this area will be. So it's kind of a how reliable is the scoring, and so the higher you set the value, you need to be more sure of your genotype call. So that shrinks this area. And then, oops, where is GenomeStudio? Here. Once you've all said that, you can hit finish, and it should be loading all your data. Now I've noticed that if you're doing this, sometimes I've noticed that GenomeStudio, when you click finish, nothing will happen, and it seems like GenomeStudio is a bit stuck there. The only solution I found for that is closing off GenomeStudio and restarting it and restart the process. If you do that, it should remember all the things you filled in before. So every time I set, this is where you need to identify where the sample sheet is, or here's where you say where the data is, it will remember that. So it's at that, check it, but at that point when you redo this, you can just more or less hit next without having to change much. And I haven't had it where it goes wrong in 2 times in a row. So I hope this is working for everybody. And if it isn't, then I hope that the second time it will work. I will wait a little bit more. So now my data is loaded, and again, that depends a little bit on the speed of your computer. So I'll just wait a few seconds to, if there's other computers, to give it a little bit of time before I start talking more about this. What we see here. So one of the things, so you have this, so if this all worked well, you now have a project in GenomeStudio that holds all the, that has all the scoring for all the individuals we identify in that sample sheet. Now, if in a month from now I get some new data in, I can either redo this whole process and make a new sample sheet and load everything in, but maybe I've already adjusted a bunch of things, or I've been working in this and I don't really want to lose the progress I made. At that point, I can just load in additional data by going through File, which is here in the upper left corner, and clicking the Load Additional Samples here. And if I do that, then really what happens is we go through the same process again as we did before, except that, well, one, you don't have to name your project anymore because it's already been named, because you're adding to the same project. And you don't have to give the manifest file, but other than that, it's pretty much the same steps as we did before to get new data into an existing project. So that would be the other way to load those two folders we had originally, rather than copying everything into a single folder, is loading them one by one into a project. And then if we look at GenomeStudio here, we see four windows or four areas. This one first, this is the SNP graph, which is right now is empty because no SNPs are selected. Now in this area here, which is called the full data table, it lists all the SNPs. And so by clicking on any one of them, I start seeing the graphs. And so you can see that the graph automatically updates whichever SNP I choose to look at here. So this area is the plots I've been showing before. So again, this tells the proportion of the B allele that is seen. This tells the y-axis indicates total intensity, and so you're expecting at least 3 genotype clusters in this graph. There's a few things we can do in this graph. For instance, we can change how it looks by clicking here on the Cartesian coordinates button, which changes this graph, and now what it shows is on the x-axis the intensity for the A allele, and on the y-axis the intensity for the B allele. And then you see that, well, the B alleles have barely any or no intensity for A, and the AB individuals are floating somewhere in the middle because they have intensity for both alleles or both colors. And then if I change this, you also have an AA cluster here, and you can already see that some individuals have a really high intensity, at least in this SNP. For here, you can see that some individuals fall in between those lines, and so they're black, which means that GenomeStudio is not assigning any genotype call or genotype to them. So if you want to go back to the other view, which I like better, is you just click on this leftmost button here, which says polar coordinates. If you click this button here, which is like 2 sheets, that copies the graph for you, so that you can copy it and then you can paste it in a Word document or whichever document you want it. I believe this button removes. The next button removes the coloring zones, but I like to have the coloring zones, the colors there. It just helps to see what's going on. And then there's some ways of selecting individuals. So I can click on an individual if you're really accurate, or you can just select a single individual and then come back to this. But below the graph is the samples table, which lists all your samples. And so you'll see that if I highlight this individual, then it will also be highlighted here, and it will show me what this individual is. Now this is a bit small, so if I want to see it bigger, I can change these columns just like I can in Excel. Now I can also, like in Excel, hold Control down and select another individual, and now this individual will be selected too. This hand allows you to move the graph any way you want. If you want to reset it, there's I think 2 ways, which is either these buttons here, which are the auto scale, so it will put the graph back to whichever, so that you can see all the samples. And then this is the lasso mode, and the lasso mode, you can just freely create any area that you want, and it will select all these individuals in the area you encircled. So that one is really useful, especially when 2 clusters come close to each other and you can still identify them separate. You can still see the 2 clusters, but with the regular box where you have a box with straight lines, and you would actually select a little bit of either cluster. If you do that with the lasso, you can really follow the contours of the clusters. And so, as I said, as you select things in here, they get highlighted in this table. I don't know where they are. They are here. The other way around works as well. You can highlight an individual if I'm interested in a specific individual, and it will highlight here in yellow. And again, also in this table, I can use control to highlight multiple individuals, and I can also use the shift. If I hold shift, then it will select everything in between the last individual I clicked and the individual I clicked now while holding shift. So it allows you to select a group of individuals if you want to do that. Also, if I select, for instance, all these AB individuals here and I go to a different SNP, I can see how they perform in different SNPs. So this table lists all the SNPs. It also gives you the chromosome, and the way for the cherry array we did it is it's because it's a cherry array, so it's not only for sweet cherry, it's also for tart cherry. We indicated by PAV that it is a SNP for the sweet cherry or the avium subgenome of tart cherry. There should be some PFR chromosomes, that means that they are targeting the fruticosa subgenome of tart cherry. They may work in sweet cherry, but they were not designed for that. And then the number indicates which chromosome it is, and in cherry we have 8 chromosomes. I'm not sure if they are. Yep, and we also added a few chloroplast and mitochondrial SNPs on this array, so that will indicate that as well. And so other than that, you can also scroll through this. Right now I'm just using my mouse wheel to scroll up and down through the SNPs, if that's a way to go quickly through SNPs. And so what else is in this table? You have the GenTrain score, which is an indication of the quality of how well your SNP was scored. So we see here one that is a bit lower, and well, you can maybe see why, because there's a lot more individuals that are not in the main clusters. And as this number really drops, then the quality of your SNP scoring really goes down. Then after some other columns on the performance of the SNP, you see for each sample it will start listing the genotype here, the score, so that's a quality score, and then theta and R. Theta is here the x-axis and R is the y-axis, so it's telling us where in this plot the individual is situated, and it does so for all individuals. And I believe, and I have to check, but so right now Giant is on top of the sample list here on the left, and Giant is also the first individual listed here. Now if I select this column by clicking on the column name, and I say I want to sort this from A to Z, as you can see now also my samples here are sorted on the same order as they are in the sample table. So these also interact in a way, as that they sort the samples in the same order. Trying to think what else I wanted to say here. So there's a bunch of buttons here as well. Again, those two sheets mean copy. If you want to export this data file, because you want to, for instance, have the genotypes, you can press this button here, and it will export it to either a text file or a CSV file. The next button here allows you to import data, which is really cool. If, for instance, I have an idea of how well all these SNPs perform, I can have an Excel file that lists all these SNPs names plus a column of performance, and I can load in that Excel file, and then it will create an extra column here that gives me, for instance, SNP performance or anything else I want to know about these SNPs. So you can add information that doesn't necessarily is automatically in GenomeStudio. Then there's a few graph functions. I'll come to those, back to those in the next session. I think one important thing to show is the filter. So you can filter here on any column in your table. I'm just gonna, so if I say, for instance, I want to know a SNP that is called, I know there's a SNP called CLF and I want this, I click on name, I say operation equals because I want all the SNPs that have this name. To press this arrow here to make it a filter criteria, and if that's the only criteria I want, I can press on OK, and you'll see that now this table is filtered for all the SNPs that are according to this criteria. Now this button here clears the filtering, and if I filter again, you will see that, well, everything that was set, it will remember that I asked for a CLF SNP, but it will be gone back to smaller than, and it will standard again have the column I want to filter on to the very first column. Now if you need to search for SNPs a lot, this might, well, always changing all of this takes a lot of extra time, especially if you really need to do this a lot. So one of the things I've been doing is using the index to filter, because it's the first column anyway. And so if you look here at the index, it's a number, and it's basically a number from 1 to the number of SNPs that are on your array. And within the SNP array, that number is unique, so number 10 will always be DDF1. So instead of using the full name of my SNPs, I just use the index because it's also a unique identifier. Now, if I want to, for instance, know now that I want to work with SNP number 667, for instance, then I can write here 667, and I can just say I want equal. And if I do that, again, I click this button to make it the criteria to filter on, and pressing OK, I will now have this SNP. I just chose the SNP at random. It doesn't seem to be a really good SNP, but that's how it works. Now if I want to use the next SNP, I still have to change this operation thing every time. So the other thing I do is I check this little mark that says not. And so by saying not, I'm saying actually filter for any SNP which has its number is lower than 1236 in this case. But by saying not, I'm saying actually do the opposite. And so right now it will be filtering for everything that is equal or larger than this number. And now if I again move this over here so that it is the filter criteria I want to use, and I press on OK, then this is the SNP. The very SNP that appears at the top is this SNP that I was looking for. Now if I remove the filtering again and I want to look for a different SNP, you can see that the not remains checked. So that's one of the only things that doesn't reset every time. So from now on, I can just type in any number I want as a filter criteria, and I can really quickly go to the SNP I want. That definitely has saved me time. It sounds like, I mean, it's just two little extra actions if you don't do this. And, well, during this curation, I looked at hundreds and hundreds of SNPs, so it really adds up in the end, and how quickly you can just move to the SNP you want. And again, this was a random SNP, and this is also not really a SNP that is performing well. What else do I want to show? Oh, this is the column chooser here in GenomeStudio. If I choose this, it will list the columns that are being displayed right now. And so it shows the index, the chromosome, and then all the samples. So I could exclude samples here if I want by saying don't show this sample. So I click on the sample, and then I press hide, and then it won't be in the table anymore. Or the other way around, if I do want them, I select the samples and I say show them. And they'll appear at the bottom of this list now. Now why is this important? Well, also you have this for displayed subcolumns means for every individual it's showing this genotype, the score, the theta, and the R. But I might not really need all this. All I need is the genotype score, for instance. So I can hide all these columns by selecting them and then pressing the hide button. So again, I select these were in here, and then I press hide so they are not shown. If I then press on OK, you'll see that this whole table is changed so that it only shows the genotypes. and there's and as we will use this in the next session to get a different statistics for each individual and just only get that statistic. Now, as you may see here on the top where the full data table is, there's also a SNP data table. So this area houses two tables I use regularly, and the second is the SNP data table, which provides more information on the SNP rather than on the samples. And so some of the data that is important is here the call frequency, which is saying what proportions of individuals got a genotype assigned. So you see that in this case it's 0. For instance, this SNP has a really good call rate because there's only one SNP that doesn't have its genotype called, and then this one has 1, so that's the maximum, which means all SNPs are called. Doesn't mean they're all called correctly, it just means they're all called. Then the PC, the PPC errors, this is kind of listing how many errors do I see in this data based on parent information I provided, or replicate. In the case of rep errors, how many rep errors in the case of reps I provided. So those are useful statistics to look at. And then if I go a bit further here, then it also gives me some more the quality scores again, such as GenTrain or the cluster SEP, which indicates how well my clusters were separated. And then all these are the AAT mean. The T stands for theta, and here at the R mean stands for it's just the R in the y-axis. So basically what this is saying is where is the average of this cluster, of the AA cluster for this SNP in the graph. So it's, in this case, if it's the BB, so the BB T mean here and the BBR mean, those give where this little plus symbol is here in the graph, which is what GenomeStudio considers the center of that cluster. And then similarly, the AAT def and the AAR def is like how much does that value deviates within a single genotype cluster, and that is represented in this graph as the width of these circles. So this would be the the BB R def, and along the x-axis, the width along that one would be the BB T def. That could give you some useful information, because if your cluster is really, really big, something might be, or at least the deviation is really big, then that might indicate something is going wrong as well. The other thing I want to point out here, I guess, is that I did a filtering. Sorry, I went back to the full data table. This is where I did my filtering on my SNP, but as you can see here, the same filtering is applied to also the SNP data table. So that means if you want to filter on certain quality scores in this table and then get the genotypes for the resulting SNPs, you don't have to redo or do any different filtering, because these two tables are automatically filtered the same way. And then if you're wondering, I filtered and I did some filtering and I want to know how many individuals did I select, or how many individuals remained after filtering. That is shown here at the bottom of the SNP and full data table. You see here display is, and then a number. This shows you how many individuals are being displayed, and it also shows you how many individuals, or how many SNPs, I have selected. So if I select a few SNPs, then now it says selected is 5, and that also works for the sample table. So in this case, and everybody should have 77 individuals in their data set. Right now, 1 is selected, and all of them are displayed. And then I haven't really talked about this part of GenomeStudio. Right now, this is empty. Let me try and see if I can find an individual. So here, now this SNP says in the SNP table there are some parent-child and parent-parent-child errors, and I'll come back to those later in the next sessions. But if there are errors, then they are listed in this part of the graph, and it will tell you what the error is. So in this case, there's a child that was BB, and one of the parents is AA. So that can't be true. So GenomeStudio says something must be going wrong here. So it gives you already an indication of if things are going wrong with your SNP. You can start looking at this table to see what's happening. It also does that for duplicates if there are any errors in your duplicates or in your replicates. The only thing is that you see that this table is cut off, and I can't really scroll right now through it, unfortunately. And if I scroll through it with my mouse, then it will just disappear from sight. So the only way I found to do that is to slightly change the size of this table by going to the edge of it and just dragging it a bit. And at that point, you can scroll. And you'll have to do that every time. Every time you choose a new SNP, and this happens, unfortunately. But I haven't found another way around it. If anybody has, please let me know, because it's a bit frustrating. The other thing I want to show here is that if I choose here an individual with possible errors, is that, well, that one, those individuals are highlighted in this graph as well, and they're not only highlighted in this graph. Because they're highlighted in this graph, they're also highlighted in this table. So everything in GenomeStudio is quite connected. I think we're nearing the end of our 1 hour and a half, and I think I've mentioned most of it. I think I realized that as we combined our sample sheets, those may still not have had the parent-child relationship. So I will upload a new sample sheet file for everybody on the Dropbox that has these parent-child relationships in them. For those who are really familiar with Cherry, you might see a few things that look weird to you. That is on purpose, because I want to show you a few principles, and so I had to introduce a few things that were not true in the pedigree, just to be able to show you in the later sessions. So I think so. Before next session, I will share that new sample sheet file, so if everybody can use that one, so they have the parent-child relationships in there, and then create a new GenomeStudio project with that sample sheet, and just make sure that everything works for you in GenomeStudio. That would be great. Other than that, I think I talked about everything I wanted to talk about today. I think the only other thing that I remember is that, so if I have this row here highlighted, and I scroll down because I want to find another SNP for some reason, and then now I wanted to actually have the one SNP above, if I would be scrolling with my mouse, with my scroll button on my mouse, and I go up, it will not move up the table. Instead, it will move up your sample, or your individual, or the SNP in this case that you have selected in this table, and so you're back all the way to the top rather than where you want it to be. So if I wanted to go to, let's say, SNP 1000 and I just missed it, or SNP number 10,000 and I just missed it, one, I could try and fine-tune it by scrolling here, or I can just click on this and then use my mouse wheel to scroll up. Because if I don't click on the sample here right now, as I showed before, then I just jump back up to where the last SNP was that I highlighted. So that's another thing that, just a little trick in GenomeStudio. I think, well, you can, other than copy, you can also save this graph by right-clicking on it. So everything, I think, is right-clickable in GenomeStudio, and then say copy image to file as a bitmap or a JPEG or whichever format I want. Oh yeah, and I forgot one more thing, is that if I highlight some individuals here, I have to press this button to copy, and then if I did it right, this should copy everything with all the columns. Now if in Excel, so what I did is, that was maybe a bit too fast. So I clicked here on an individual I want the data from, I went to these double sheets here to copy it, and then I went in Excel and I pressed Ctrl V to copy, to paste, sorry, and I got all of that. Now if I only want, for instance, the sample name and the short name, I can highlight both by, well, I highlight one by clicking on the name of the table, then I hold Control and I click on any other table I want. Now if I copy this, and if everything went well, and I paste this using Ctrl V, then I only copy the columns I selected. And so that is useful if I want to manually adjust a group of individuals. Let's say I want to, all these individuals, I want to make them AA, and I can't readily do this necessarily in GenomeStudio. Well, they're now all selected in this table. So I hit copy again, and you have to hit copy because Ctrl C has never worked for me. And then now I've pasted all these individuals, and now in Excel I can start making notes on these, maybe call these AA if I want to. And that reminds me of one more thing. In, let's say here in the SNP that doesn't have a lot of calls, but I'm gonna filter it. I'm gonna remove the filtering because I think there was one on the top that worked better yesterday. Maybe not. So here I have two clusters, and they're both in the AA realm. Well, maybe I know that this should be AB and this should be AA. So I can tell GenomeStudio by selecting a group of samples here in the SNP graph. And then right-clicking on them, I can say define AA cluster using selected samples. And then it will, in this case, it won't do much. So in that case, you really have to go in manually, select all of these, go in Excel and call them. In some cases, that will work, and GenomeStudio will correctly identify everything after that. So yeah, so again, that is selecting the group of samples you want. I'll try a different one. And then saying this is actually the AB cluster. And then GenomeStudio will determine what the other clusters are, or which other clusters it can see, and do your SNP calling. Okay, I think that will be it for today. If I forgot something else, I will come back to it. Next session, in the next session, well, we'll start talking about the rest of the workflow. So next session will be mostly about removing any individuals that have a low quality. So sometimes we get individuals data back, and we can almost straight out see that that data is no good, and so that we don't have any trust in that. So in that case, we'll have to rerun the individual. So I'll show you how we can identify those. I will also show you how to determine diploidy, and in case of apple and cherry, sweet cherry at least, we're expecting diploids. So anything that is not is doing a non-diploid will throw us off later on. So we remove those for now through the workflow at least. And I will show you how we get a subset of these SNPs that are reliable, rather than just going one by one through all the SNPs to see which one is working and not. There's some software that can help us do that. So that will be for next session. And so as I said, I'll provide a new sample sheet, make a new GenomeStudio project with that sample sheet that holds the parent information, and then play a bit around in GenomeStudio, see how everything works, and get yourself familiar a bit with it. And so, yeah, that's it. And so the next session I plan to have next Wednesday for people in the US. That's at the same time as it was today. And then for everybody in Europe, it's going to be an hour earlier because the US switched to daylight saving times by next week. But I'll send out another reminder email about this. So I hope this was all clear. Yeah. Are there any questions for now, for today? Nothing? Okay, then I think that we'll end today. If you have any more questions, if you go over this through the slides and you're doing it yourself and you encounter some questions, then please let me know and I'll try and help you as best as I can. And I'll see you all next week.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Bye.
Thank you. Bye.